So, very good morning to everyone. How are you, children? Am I audible, visual, everything all right? Give your input first. Audio is okay. Screen is visible to you. Everything all right? Fine. Give your input. Then we will start the today's session. Right. So, first information I just want to give you, from the next class onward, we are starting our track sheet. So what are the best way? You start reading from today itself, and every good student do one good thing. Read the topic before listening the lecture. So we are going to start with morphology in next class, and we will deal about root system. Root system and root system. So make your preparation for the root system. You read through the line of NCERT. It is hardly one page. You can manage it easily. We will discuss it during the time. Got the idea? Now, today we are going to discuss the paper. Don't think like it is a waste of time. I will tell you a lot of information related to question while discussing the paper. Got the idea? So don't think like I already know the paper. I have already answered the question. I know everything related to it. So what is the use of it? It will help you, don't worry. Okay. So now we are proceeding about the paper discussion. Voice is breaking. It is for all. And I will also say you while discussing these questions, how to solve the bits. So I think two people told voice is breaking. Is it for everyone? Sound is not very clear. Hello, hello, hello. Audibility is okay, okay. Good. Right. Now give your attention. So we are starting the today's session. In this particular set of the question, the very first question is, identify the set of correct statement. This is a statement type question came in it, right? And they are asking the set of correct statement is what? Now, the very first statement here is, the flower of Vallisneria are colorful and produce nectar. Is it a true statement or false statement? So, can you give your input? First one is a which one? False statement. Why it is a false statement? Vallisneria is an aquatic plant. And as you know, in aquatic plant, Vallisneria especially, pollination occurs by water. Aquatic plant not the guarantee of water pollination, but in Vallisneria, pollination occurs by water. And water is a abiotic pollinators. What is the need of nectar in such particular plant where water is a pollinating agency? Nectar are there used as a floral reward when biotic agents are there, when insects are there. Here pollinating agency is water. So what is the need of the nectar? And that is the reason this statement become a wrong statement, incorrect statement. First statement become an incorrect statement. Okay, ji. Now we are coming on the second statement. The flower of water lily are not pollinated by water. Means they are telling in water lily, pollinating agent is not water. It is a true statement. If you read the NCERT, they wrote in all aquatic plant, in all hydrophyte means the plant who grow in the water, water is not a pollinating agency. And two example NCERT wrote, one example wrote water hyacinth and another example they wrote which one? Water lily. So in water, whose name start with, I am just telling for making you mug up. So to make it easy to mug up, whose name start with water? Water hyacinth, water lily. They not get pollinated by water. They are aquatic plant. But NCERT wrote that water lily, water hyacinth are aquatic plant, and in these plant pollinating agents are winds or insect, but not the water. So of course, the second statement become a correct statement. The flower of water lily are not pollinated by water. Second is a correct statement. Now, third one. In most water pollinated species, the pollen grain are protected from wetting. Of course, when it become wet, it become heavy, it will sink. So pollen grain are covered by a hydrophobic coating, so it protects it from getting wet. It is also a correct statement. Fourth statement is saying that pollen grain of some hydrophytes are long and ribbon-like. A statement, of course, correct. It is written in NCERT, but I am asking a question to you people. What are the name you will write in place of some? What are the name you write in place of some? A statement is correct. What is the name of hydrophytes whose pollen grain are ribbon-like and long? Write the name of that plant, hydrophytic plant. They wrote some hydrophyte. I am asking name of that hydrophyte. Please answer it. What is the name of these hydrophytes? I am waiting for your response. First try to answer without help of NCERT. One answer came, Vallisneria. It is not a correct answer. Sorry. Next answer. Water lily, not a correct answer because water lily, got the idea? Pollinated by, not pollinated by water here. So answer here is Zostera. Seaweeds, Zostera. Getting the things or not? Well done. So in examiner can ask another question, na? In Zostera, pollen grain are long and ribbon-like. And I am telling you an interesting fact also here. Zostera have reported longest pollen grain. So anybody going to ask longest pollen grain present in which plant? What the answer will you give? Zostera. Got the idea? Clarity came. Now for next statement is, so D is also a correct statement. In Zostera, it is a hydrophyte where pollen grain are ribbon-like. It is correct. Now we are coming on E statement. We are just looking either correct or incorrect first. In some hydrophytes, the pollen grain are carried passively inside the water. Of course, it is also a correct statement, but I am asking once a question to you, what you will write in place of some? In which hydrophyte? In which hydrophyte pollen grain are carried passively inside the water? What is the name of hydrophyte? In NCERT it is mentioned. In NCERT, water pollinating plant, how many example is mentioned? 2 example is mentioned. One mentioned hydrophyte is Zostera. Zostera is an example of hypohydrophyte. In Zostera, pollination occur under the water. Got the idea? It is under the water pollination occur in Zostera. And in case of Vallisneria, what happened? Pollination occur on the surface of water. So suppose that here is the stigma. here is the stigma, and here pollen grain release. So along with the water current, pollen grain move near the stigma and pollinate the flower. Got the idea? Is it clear in the mind? So getting the idea, is it clear in the mind? Understanding came, clarity came, right. So it is also a correct answer. So in Zostera, pollination occur under the water. In Vallisneria, pollination occur on surface of water. So these are the some of the information we also gathered from this question. Now we are moving toward the next question. Just have a look. Now we are coming to next question. This is the question from ecology, and in Andhra Telangana state syllabus, ecology is not in botany, it is in zoology. In entire India, ecology in botany. So I am not taking this question. I am coming on next question, 103 question. Got the idea? It is in zoology, botany section in your state. So we are going to talk about this question. Here question is inhibition of succinic dehydrogenase enzyme by malonate is classical example of. It is a easy question, but I am asking from you people, have you idea about these process? Where these inhibition occur? Where these inhibition occur and what is the need of these inhibition? So I hope all of you are aware about the Krebs cycle. Okay, ji? If we talk about the some of the important steps of Krebs cycle, you are finding in Krebs cycle what happening? Oxaloacetic acid react with acetyl coenzyme forming citric acid. I am telling NCERT things. Citric acid converted into alpha ketoglutaric acid. Alpha ketoglutaric acid convert into succinic acid. Succinic acid convert into fumaric acid. Fumaric acid convert into malic acid, then convert into oxaloacetic acid. I hope everyone know about these reaction. Is it all right? Is it all right? Is it clear? Done? So suppose that in a cell for a time being there is no need of Krebs cycle. In a cell, suppose that for a time being there is no need of which one? Krebs cycle. Cell don't want to perform Krebs cycle for a moment. Means cell want to stop the Krebs cycle to occur. How cell will stop the Krebs cycle to occur? I am telling you, answer is hidden here. So I am explaining this answer. How cell do it? So actually the normal reaction is, I am just telling you, this is succinic dehydrogenase enzyme. And here substrate is, in short I am writing succinic acid. This is substrate. Now what happened during reaction? These succinic acid substrate bind at the active site of succinic dehydrogenase and forming a product called fumaric acid. It is the product. So this is a reaction, normal reaction. In normal reaction in Krebs cycle, what happened? Succinic acid bind at the active site of succinic dehydrogenase enzyme. This enzyme is present in inner membrane of mitochondria. Getting the things or not? How to stretch information from a small thing also, you know it. So see, succinic acid bind with succinic dehydrogenate converted into which one? Fumaric acid, normal reaction. Suppose that cell don't want this reaction to occur. What option cell have? So cell have a option. If cell want to stop the reaction, they will start supplying in mitochondria which one? Malonic acid. These malonic acid also called what? Malonate. In biology, we say citric acid as citrate, malonic acid as malonoate. Now what happened? Try to understand. This malonic acid is an inhibitor. Suppose that this is malonic acid. It is an inhibitor. Inhibitor is also called substrate analog. Inhibitor is also called substrate analog. It is similar to substrate. Now what happened? Inhibitor concentration start increasing. As the concentration of the inhibitor become greater than substrate concentration, that is succinic acid, this inhibitor will bind at the active site. This inhibitor will bind at the active site. Now inhibitor is attached here. Got the idea? So when inhibitor bind at the active site, means who is fighting together? Substrate and inhibitor are fighting together to bind at the active site. Competition occur. Active site is one, and how many contestants are there? Two. Both are fighting together. They are telling I want, they are telling I want. Who will want? Who is more powerful? Who is more powerful? Whose concentration is more? When the concentration of inhibitor become greater than the concentration of substrate, it will bind here, and as long as it will remain attached, who can't bind? Substrate. Who will not form? Product. Reaction come on a hold or not? Reaction stop or not? Formation of fumaric acid stop or not? Now you can ask, sir, once again how they will start? How they will start once again the Krebs cycle? One cell want to continue Krebs cycle, they will increase the concentration of which one? Succinic acid, which become greater than which one? Malonylic acid. Now when this is going to bind, the both will fight together. If and in fight whose concentration is more, they win, they are the winner. Once again reaction start. So this question is based on this concept. This question is based on this concept. I told you about the concept of which one? Competitive inhibition. I hope you understand it. Both substrate and inhibitor are competing, fighting together for active site. What the idea? So inhibition of succinic dehydrogenase enzyme by malonyl is a classical example of which inhibition? Competitive inhibition. Got the idea? Right? No any example of feedback inhibition is given in your syllabus. I am not going to decode all the option here, otherwise entire class will finish to decoding this question only. Right? So we will teach when we are going to talk about the enzyme, then at that time I will tell you also what is feedback inhibition. Is it clear? Now we are moving toward the other question. So I hope you understand about the competitive inhibition. Now the next question, of course, a easy question, but I know many students did it wrong. And many students did it wrong. Even I told you even student who scores more than 700 mark also did it wrong. One or two students. Our student only telling. And what is the reason of doing wrong? I am telling you. What they told after coming from the paper, getting the things, examination hall, I am just telling you. The question here is, identify the part of seed from the given figure which is destined to form root when the seed germinate. Here the diagram of a seed is given. I am labeling it proper. I am just highlighting it properly so you can understand proper labeling. It may be possible paper is right now there with you. So here one thing is this, another thing is this, and labeling is here. You better know, everybody know, root arise from radical. And the root who arise from radical is taproot. In dicot plant, generally root arise from which one? Radical. Every student know. These are the basics of the morphology when everywhere we study. Root arise from radical. Then you can ask then why students are making it wrong, sir. So the outer red color, or I am changing the color so you can differentiate better. I am changing the color so you can differentiate better. This red color one is which one? Coleorhiza. And this blue color labeling is which one? Radical. Getting the things. What some of my students told, ki, sir, this labeling is, this arrow is not touching the radical. That's the reason I get confused. Actually, which one is the radical? This blue one is the radical. They are telling this arrow is above that blue, sir. That's the reason I get confused. Now I am asking the same question to you. How many of you did like this? Of course, this is the mistake of paper setter. They have to point out the very correct labeling to not confuse the student. But anyhow, you can see like two lines are there. One is coleorhiza, another is radical. If it is coleorhiza, then this becomes which one? Radical. We have to say like this. Nothing, anything is getting the idea. So radical is giving the root. So we have to consider it is radical. But some problems was there from paper setter side. They have to point out the arrow in a very appropriate place. Without it is a competitive exam and you don't leave any glitch where a student get confused. Now some student also raising hand here ki I also did this mistake due to this reason, sir, because it is not very properly pointed. Because it is not very properly pointed on the radicals. Got the idea? Right? So it is getting the things. So certain time we have to use some common sense. See now, if two things are there like this, one is coleorhiza, another become radical, and they pointed here. So student confusions are there. This is the reason, getting the things. So, okay, don't worry, it happened. It doesn't mean your knowledge is less. It getting the things or not. And I will say also, it's possible, quite possible. Who made it mistake? I cannot say that a student don't know this. They know this, but anyhow it happened. You can say it is a bad luck. Got the idea? Now we are just moving on the next. So here bulliform cells are responsible for. First question I am asking to you, out of dicot or monocot where bulliform cells are present, answer it. We are going for a small revision. You can say like from next day we will go for the track sheet. So this is the last class of such kind of thing which is haphazard, not very planned, getting the things or not. So bulliform cell is present in monocot. Very good. One answer you have given. Adaxial or abaxial surface where it is present? Bulliform cells are present on adaxial surface or abaxial surface. Answer it. AD or AB? Adaxial surface. It is also a very good answer. What are the characteristic feature of bulliform cell? How many character NCERT wrote? NCERT wrote it is large. It is empty. Or it is colorless. It is written or not written? It is large, it is empty, it is colorless. It become turgid when water came and it get expanded. It become flaccid when water scarcity is there and the leaf is going for inward rolling. Why the leaf is going for inward rolling? To protect the water loss. When it rolling of leaf occur, leaf get rolled, what happens? Surface area of leaf will decrease and water loss will occur less. Got the idea? So to reduce transpirative loss of water. Got the idea? Is it clear? So this is the question. We got the information about some more information about bulliform cell also. So bulliform cell present in monocot, it is present on adaxial surface. It is large, it is colorless, it is empty cell. It become turgid when water is present, become flaccid when water is absent. When it become flaccid, it going for inward rolling. So there are the options. Of course, the answer is what? Inward curling of leaf in monocots because it is not present in dicot. So I am asking a question. If a bulliform cell is not present in dicot, what kind of adaptation is there in the dicot to reduce lot of water? Tell me, answer it. If a bulliform cell is not present in dicot, Then what kind of adaptation a dicot have to reduce the loss of water? Who will answer it? I am waiting for response. Monocot, it did the curling. It reduced the surface area. What dicot is doing to reduce loss of water? What adaptation is there in the dicot? Here curling occur. There, one student wrote stomata. So stomata is not present in monocot. Stomata present in both dicot and monocot. Na? So in dicot, two things is there. One, hypostomatic stomata. More stomata present on the dicot leaf is dorsiventral leaf, like this. So in dicot, more number of stomata present on the lower surface. So this part is in shade, this part in sun. So the part who are in the shade have more number of stomata, getting the things, or the part who are in facing the sun have either less number of stomata or stomata is absent. And here thick layer of cuticulization is there. Here thin layer of cuticulization is there. Getting the things or not? So these are the adaptations present in the dicot leaf to preserve the to reduce the transpirative loss of water. Okay, this question is over. Some left right we have also seen here. Now we are going to move about the next question. Here the next question is, which of the following one required for a dark reaction of photosynthesis? Light not required, it required in light reaction. You better know. Chlorophyll not required, it required in light reaction. CO2 required in dark reaction. ATP also required in dark reaction. NADPH also required in dark reaction. Got the idea? Now, this question may possible to make it more difficult. If I ask a question in place of NADPH, if we wrote NADH, then the option become correct or not? NADPH I not wrote. I wrote here NADPH. Then answer becomes CDE, or then the answer become different. Tell me. Different. Because NADP required during respiration. NADH required during respiration. pH required during photosynthesis. If NADH anybody have given, then we not count these options. So correct answer, because NADPH is there, so correct answer is C, D and E, and based on that we did it first option. Now I am asking a question, other question. In a dark reaction, in which step CO2 require? Which step of the dark reaction requires CO2? Pin your answer. Pin your answer. Dark reaction, if you are confusing, I am helping you. Dark reaction means we are talking about C3 cycle. Synthesis of sugar occur in C3 cycle. So if you are confusing, now I think it become easy for you. Na? At which step of the dark reaction CO2 require? One correct answer came, carboxylations. Good, well done. Now at which step ATP require? ATP require in one step or more than one steps of the dark reaction. So ATP require in two steps of dark reaction. One is regeneration, another is reduction. Reduction and regeneration both require ATP. NADPH require in which step of dark reaction? Reduction. Got the idea? So people can frame question of such kind also from here. CO2 require in which step, ATP require in which step, NADP require in which steps. Got the idea? CO2 require in how many steps? ATP require in how many steps? NADPH2 require in how many steps? People can change the things and make the new questions. You got the idea? So this is another one. It is also done. Right? Now we are going to deal the next question. The next question is formation of interfascicular cambium from fully developed parenchyma cell is an example of. Now leave the answer. Answer already circled. Answer already highlighted. So nothing is hidden in the answer here. My intention is also not to say you the answer. Once we already discussed this paper, but beside I am discussing. But the intention is to say in the periphery. Listen it carefully. So what I am saying, formation of interfascicular cambium. Now I am asking a question. Interfascicular cambium is which type of tissue? I am giving you option. It is primary meristem, it is primary permanent tissue, it is secondary meristem, it is secondary permanent tissue. Interfascicular cambium is which type of tissue? 4 options I have given you. So interfascicular cambium is which type of tissue? Primary permanent, primary meristem, primary permanent, secondary meristem, secondary permanent. So it is, answer came from your side and a very good answer came. You are telling, sir, it is secondary meristem. Interfascicular cambium is secondary meristem. They are telling interfascicular cambium, formation of interfascicular cambium from fully developed parenchyma. Means parenchyma converted into interfascicular cambium. Here it is parenchyma. And this parenchyma converting into interfascicular cambium. I am writing here parenchyma. Wait, parenchyma. And here I am writing interfascicular cambium. You better know parenchyma is a which type of tissue? Parenchyma is a which type of tissue? Simple permanent tissue or primary permanent tissue. Primary permanent tissue. It is a primary permanent tissue. And interfascicular cambium is a secondary meristem. It is a secondary meristem. Conversion of secondary meristem from primary permanent tissue is called de-differentiation. Got the idea? What is differentiation? When primary meristem converted into primary permanent tissue, this event is called differentiation. So in this question, all definition is over. What is differentiation? Conversion of primary meristem to primary permanent tissue is called differentiation. What is de-differentiation? Formation of formation of secondary meristem from primary permanent tissue is called de-differentiation. And when secondary meristem converted into secondary permanent tissue, it is called redifferentiation. There are 3 terminologies there. So primary meristem to primary permanent tissue, differentiation. Primary permanent tissue to secondary meristem, de-differentiation. Secondary meristem to secondary permanent tissue, redifferentiation. This question is also over. Now we are, so answer is which one? De-differentiation. It is already highlighted. Now we are going to talk on the next question. Right? So this question, where is this? Just a minute. Now here it is a question number 108. See, exactly NCERT not wrote about it, but we can guess it. Got the idea? Exactly, NCERT not wrote ki hind 2 have how many base pair? They have written about which one? Eco-ARUN. And usually they are writing palindromic sequence or 6 base pair example they have taken. In eco-ARUN, who is there? GAATTC is there. GAATTC is there. Got the idea? So it is how many? 1, 2, 3, 4, 5, 6 base pair. So they are writing hind 2 always cut DNA molecule at a particular point called recognition sequence, and it consists of. So usually most of the recognition sequence, that is also called palindromic sequence, are made up of 6 base pair. Most of, not all. Some palindromic sequence are also there who have 4 base pair only. Getting the things or not? But NCERT have given the example of 6 base pair in which one? Eco-ARUN case. So we can copy paste it. So here that small risk you can take. Getting the things or not? You can take the benefit of doubt because NCERT not told, and about eco-ARUN NCERT author told, vaya 6 base pair is there. So we are just going to tick it which one? 6 base pair. But how many base pair is there in the hind 2? They not wrote. It is also 6 base pair, but it is not mentioned in NCERT. We just connect it with the answer of which one? Eco-ARUN. Done? Over. Can we move ahead? Now we are moving ahead. Next. It is zoology in your state syllabus. Now we are coming on this question. It is in botany. It is easy question. And right. Next question is, which one of the following is not a criteria of classification of fungi? First option is mode of spore formation. It is a criteria. It is a criteria. We know, you also know, I also know, but I am asking a question. How it is a criteria? How we use the spore formation in classification of fungi? Ascospore, basidiospore. Where ascospores are there, they are ascomycetes. Where basidiospore are there, it is basidiomycetes. Done. So mode of spore formation we are using. Second, fruiting body we are also using. Basidiocarp, ascocarp. Basidiocarp is the fruiting body in which one? Basidiomycetes, ascocarp in ascomycetes. So we are using it. Morphology of mycelium. Aseptate mycelium present in Phycomycetes. In other 4 groups, septate mycelium is present. We are also using it. Phycomycetes have aseptate mycelium. Remaining other 3 have which one? Septate mycelium. What we are not using in the classification is nutrition. Fungi have saprophytic mode of nutrition. They are saprotrophs. So we are not classifying the fungi into different group based on the mode of nutrition, and that is the reason it not become the answer. Based on it is the answer, correct answer. They are asking which one? Not. So if they are asking not, what we are not using? Mode of nutrition we are not using. Done. Simple question. Almost simple question is there. You cannot say anything was a rocket science ask. But accuracy is important. Even in simple exam, a lot of commitment required to come at which one? 360 type of a score. And simple exam is more, I told you in last class also, simple exam is more dangerous. If difficult exam came, it is good. You will find chances of selection is more in the difficult exam rather than in a simple exam. Listen. So what I am telling, next question, how many molecule of ATP and NADPH are required for every molecule of CO2 fixed on the Kelvin cycle? You better know. Now I am asking question. My intention was not to say about 3 molecule of ATP and what? 2 molecule of NADPH2. I know every student who are sitting here know it. I am asking 2, 3 related small easy question. Where 3 molecule of ATP require? First answer it. Easy, difficult not there. Where these three molecules require. Two in reduction stage, one in regeneration stage. ATP done. Where NADPH require in reduction stage. So who needed more? ATP needed more than NADPH. Agree or not? ATP needed more than NADPH. Now I am asking a question. NCERT have given the answer of this question. From where these extra ATP came? During photosynthesis, during Calvin cycle, my question is, I am summing up my question. During Calvin cycle, the number of ATP requirement is more than number of NADPH requirement. Agreed? So from where these additional ATP came? ATP requirement is more, NADPH requirement is less. In non-cyclic photophosphorylation, both ATP and NADPH produce. NCERT wrote additional ATP probably came from G. Iswar wrote a correct answer. Cyclic process. NCERT wrote, you were able to connect with that line of the NCERT. The additional ATP required came from which process? Cyclic process. Is it clear? And it is a correct answer. Okay, that a very easy question. It is done. My intention, I told no, but not to deal it. Now, it is ecology. It is 113 question. So this question is from which chapter I am asking now? According to new NCERT, this question is from which chapter? Who will answer the name of chapter? Question number 113. In old NCERT, it was in strategy. That chapter is deleted. Then why they ask the question from deleted chapter? Now it is in application of biology. So every part of tissue culture strategy is deleted. But what author did? They removed some topic of a strategy and shifted into which one? Application of biology. Got the idea? When you read application of biology in agriculture, their tissue culture is mentioned, their somatic hybridization is mentioned. So tissue in a strategy variety name they remove, but tissue culture and somatic hybridization is still there in the different chapter. That is what? Application of biotechnology. The capacity to generate whole plant from any cell of the plant is called, totipotency is the correct answer. Now I am asking, what is micropropagation? Answer it. What is micropropagation? Totipotency is answer, no problem. You know about it. Potential ability of any plant cell to form a whole plant is called totipotency. What is micropropagation? Producing thousands of plants from an X plant during tissue culture is called micropropagation. What is differentiation? Process of formation of primary permanent tissue from simple meristem is called primary meristem is called differentiation. What is somatic hybridization? Fusion of two somatic cells. An example, potato and tomato is called what? Somatic hybridization. So we got idea about the every options. Got the idea? So this question is also done. Right? Now we are going to deal the next one. It is zoology one. Next is botany. In your state, it is in botany according to NTA. All are botany questions, but in your state, the ecology taken care by zoology people. That's the reason I am not telling it. Now, here. A spindle fiber attached to kinetochore of the chromosome during, you know it better, this is the chromosome, this is the disc shape structure, kinetochore, from here who is coming? Spindle fiber coming and joining here. That event occurred during which one? Metaphase. Got the idea? In anaphase what happened? sister chromatid divide and move toward opposite pole. This spindle fiber pull it up, this will pull it down toward opposite pole. Got the idea? So this event occur in metaphase, and what event I am writing here, pulling is occurring in anaphase. Got the idea? It is also done. So correct answer is metaphase. Now it is a diagram based question. Identify the type of flower. So this is which one? In position of ovary. Both have a cup shape in whorl. So here it is such kind of shape here both are forming. It is which kind of condition? Perigynous. Half inferior ovary is there. Both the diagram associate with perigynous. Both have the NCERT diagram. Nothing more to say here. Now, I told in the beginning na कि भाई examiner want to check your patience. See, most of the questions here, this is a question, totally it is a factual question. No brain required, no IQ required, no intelligence required, simply patience required. If you revise it properly, you will not make a mistake. It is factual question. By Rhizopus is what? Braid mold. Agaricus is what? Mushroom. These two match giving us the answer. Got the idea? These two match giving us the answer. Getting the idea? Is it clear in the mind? So, you make it sure this year that I will never ever make a factual mistake. And it is only and only possible when you are revising and keep revising. See, consistent approach required. Nothing any big thing is there in the exam. Got the idea? Nothing any big thing is there. And why I am saying these things, consistent approach required. If you are really serious, I just want to become doctor. You are working some basic things. No one will stop you. It not require a big hard work, big effort. It requires small thing. When you are listening the class, listen it very properly. Either the things you know or don't know. If you know what happened, revision happened. If you don't know, knowledge came, information came. So two things will happen. Even you are knowing, okay, gee, I know this chapter, what big is this? Revise it. Listen it carefully. After listening, you listen carefully, ask the questions. If you are in the offline batches, asking question is more easier. While teaching, sir, I not understand it. Here I have the problem. You just once again, please explain it. Ask until unless your doubt not get clear. Don't come in peer pressure. Here, most of the students are not asking, why should I ask? If I have a problem, I will ask. Now, why other will ask? Ask the question. Write the notes. I find students do some small mistakes. They are finding your sir is writing NCERT only. Na? Why sir will write anything else when it is not coming in exam? They will write NCERT information. But they will write NCERT information in some easy language, some point by manner, some explained way. NCERT is highly condensed. So write the notes. Many times what's happened, when you are listening the class and not writing, our brain is highly active. You are sitting in the class, your body is in the class, but your mind is somewhere else. To stop it, notes writing is important. When you are writing anything, your brain not go anywhere else. You have a full concentration on writing the context. Alert, remain alert in the classroom. Don't drowsy, teacher is teaching, I am somehow drowsing, somehow sleeping. Be alert. These are some of the basic things you do now. Everything becomes great. Before starting the class, one day before, read that particular lecture topic. Is are going to teach next class route. I will finish the route and then I will sit in the class. After the class, revise the notes first, then revise the NCERT once again, then solve the bits. These are the things required. Solve the P by Q. Nothing any rocket science is there. And when anything happen plus and minus in your motivation, you think like, if I prepare well, I am going to save at least 1 crore of my parent. If it is private college, it is almost 1 crore. Even respect will not come. Be a person of a spine. Your parent purchase a seat for you. What great in this? Don't run from anything. Everywhere so much bigger line is there. So what you did with a great effort, it will come. Right. So every day when you are finding ki bhaiya I don't have motivation, you think like na 1 crore. It is around 8 lakhs rupees per month work you are doing. You think like if my salary is 8 lakhs rupees, I will do that negligency. Saving 1 crore is much difficult than earn 1 crore. Getting the things or not? If your parent investing 1 crore, they are able to invest 1 crore on your private colleges, only it is possible when they earn 4 or 5 crore, because expenditure is also there. Na? Got the idea? So when you get demotivated, think about this. Ki bhai, these I have to save every month of my parent. And simply if you are working, trust me, nothing is difficult. What happened this year, beside of that I am telling, what happened is not good happened. But beside of that, many of you are going, of your friend are going to medical college now. What level of cheating, blah, 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 blah happened this year. Someone telling paper leaker, someone telling what happened, believe happened. Supreme Court is hearing it. Exam will not going to probably 99.9. They already told only 1500 will write the exam. So don't wait for all these. What another thing came from Supreme Court. Whatever came, not getting the things or not, you just start your preparation. Don't waste your time. Don't waste your time. That is most important things. Got the idea? Make a list of these factual information. Revise every day. These type of factual information. Example. Make a list of all example. This example belongs to what? If example based question will come, I will not make a mistake. Make such kind of commitment from yourself. Everything become great. Now we are coming on the next question. Time is there or time up? So next question, probably it is a question of test cross. Next question is a question of test cross. What paper setter did? They make small changes. What small changes they did? In place of violet, they did it black. Violet flower converted into black seeded. It is a question of which, when we want to know the genotype of a dominant trait, we cross with which one? Recessive. So who is the recessive trait? Small b, small b. Suppose that we have a black color seed plant. We don't know the genotype. We know black is a dominant trait. So we cross with white. Two possibilities there. It is test cross. I'm writing in the format of test cross. White is what? Small b, small b. Small b, small b. Black is how many possibility? Two possibility. One possibility capital B, capital B. Another possibility capital B, small b. When this cross will occur, all black will come. When this cross will occur, white will come in 50-50 ratio. So when black and white coming 50-50, it is heterozygous. When only black is coming, it is homozygous. So this is the cross we will do with the white. It is a question entirely based on which one? Test cross. Got the idea? Done? Okay. Understood? Clear? So, see, I started discussing paper not in the purpose or intention of we are going to deal the question. You know, I know many of you what is the need of, see the question. If you are mistaking this type of question, it is a crime. Na? Simply information. What great is there? Nothing great is there. Simply if you revise, your commitment is needed. Once again, in last time saying, NEET is an exam who require simply your hard work. Trust me, it not require a lot of brain. If you are really hard working, even your score in this NEET, many people see, if we read, we know. If we don't read, we don't know. The simple thing, na? It is easy question for whom? Who revise it. Who not revise it, for them it is difficult question. How they know, bhaiya, Saccharomyces cerevisiae produce what? It is easy for whom? Who revise it. So who not revise it, it may be your score is 200, it may be 100, it may be 300, it may be 400, it may be 500, 550, whatever the score is there. Getting the idea or not? Even it is nothing you know, and if you are working really hard for 8-10 months, you can achieve at any level. You can, if it is possible to, you will get your seat. So don't worry about it, ki what marks I got. What marks you got, it is due to XYZ reason. Your commitment was missing, what guidance was missing, what problem is there, I don't know exactly. But even if you are at 0, if you work for the 10 months, you can achieve the result. It is not that much big. Simply commitment required, nothing big required, honesty required. Got the idea? And don't give up in the life. I told even in 1 attempt, 2 attempt, 3 attempt, whatever is it, for someone 4 attempt, if you are going to become a doctor, nothing better than this. Because in future, with the darkness in the career, what happened? I will do our graduation, I will go for civil services. When you are trying to become a doctor, no any other smaller thing will attract you. What is the problem? Who try to become doctor and just finding I am just giving it due to this subject, that subject. What option is there? You are either going to crack state PCS or country civil services. There also a lot of fight is there. Got the idea? So, and another small exam is not going to attract you. It is not looking glamorous to you. Getting the things. So just work, believe in working. And if many are cracking like you are also equally potential. They are not coming from the moon and Mars. They are from in between you. Simply anybody did who work good, who work hard, who just manage themselves in a better way, manage to deal it. Don't worry, mistake happen to everyone. And parents who are listening to them, I am also saying, just trust your kids. Don't demoralize for a small thing. It happened with everyone. So trust them, let them work, and everything become great. Okay, Ji. So see you with the track sheet in the next class. And my intention of discussion is how to handle the question, how to deal the periphery of the questions. That was the intentions of the today lecture to tell you. Right. Thank you very much for your patience, for listening. Bye-bye. Have a good day to all of you. Bye.